Our next video is on the pentose phosphate pathway, which we often abbreviate. If I can write PPP, pentose phosphate pathway. Okay, so this is another important synthetic pathway for carbohydrates. All right, we're looking at this area that I've circled on our metabolic pathways map, and we're going to zoom in on that and take a look. So when I look at this, right here, starting with glucose 6-phosphate, it's going to be the start of this pathway. And the first phase brings us down here to this ribulose 5 phosphate. Notice that, or remember, that the glucose 6-phosphate can go in lots of directions, right? We can go down through glycolysis, we can gluconeogenesis, back up to glucose, we can do storage, lots of routes. And now this is another route for that glucose 6-phosphate to come down here. And then the second phase of the PPP is this non-oxidative pathway right here. Okay, so in the oxidative portion in phase 1, which is the oxidative portion, we're making NADPH. All right, in the non-oxidative portion, we're converting among different monosaccharides. So we're converting from one to another. Pentose phosphate pathway, so pentose, so you're talking 5 carbons. Just to highlight one important piece of this, right, is that these are precursors for our nucleotides, or they can be. So from here we can make our way over there. Okay, so some key points. We already said that we are going to get from this the NADPH and these 5 carbon sugars. So we looked at NADPH just a little bit before, and it is typically the carrier for biosynthetic pathways. What we have been really looking at up till now, usually it's been NADH because catabolism generally uses that for its electron carrier. But this NADPH is generally the electron carrier as we're synthesizing these molecules. And then, as we just said, the 5 carbon sugars are the precursors for those nucleotides. We looked at that on the map. So if we go back up here and look, we talked about the fact that glucose 6-phosphate, which is the starting point for the pentose phosphate pathway, can also go in all these other directions. And because of that, an important piece of this, all right, is that glycolysis and PPP are coordinately controlled, right? The control of those has to be tied together. So what we have here, and I know this is little text, but we're going to look at these pieces in more detail. At the top part, we have phase 1. Okay, that first phase is the oxidative, so we've got a redox reaction going on. Let's write that, redox reaction. Okay, and we're generating that NADPH in that first part. So if you go back on the metabolic map, that's the part we highlighted in pink. All right, the second part, all this part in yellow. Okay, we are interconverting among a variety of sugars. Okay, so phase 1 making NADPH, phase 2 we're interconverting those sugars. All right, to get right here, there's going to be 3 enzymatic steps, okay, in that first phase to get us down to the ribulose. Here, let's, hold on, let's look at it up here. So 3 enzymatic steps to get us down to this ribulose 3 ribulose 5-phosphate. And if you look at this, this is a ketose sugar. Okay, we've got that right here, this ketone piece. And what we need is the aldehyde sugar. So right at the end of that oxidative part, we have an isomerase enzyme that will convert that. Okay. These steps, importantly, in the non-oxidative portion, are reversible. Okay, so whether they run, the direction in which they run is going to be controlled by the concentration of the various metabolites. So it's really controlled based on cellular needs. We're not going to look at all the enzymes in this phase, in phase 2, but we are going to look at situations where they would run one way or the other. If we look at a summary of the reactions in phase 2, okay, you do not need to know all of these names, okay, or the details of the order, how these all react. We're going to look at this kind of in the big picture. Just for one interesting piece, this molecule right here is actually the precursor of histidine, okay, this 4-carbon molecule. And another interesting piece is, notice at the very bottom, if we carried this out all the way down, which sometimes we will, sometimes we won't, but we would end up with these fructose, these 6-carbon sugars, all right, and the GAP, this 3-carbon sugar that we've seen before. And this would be our net reaction. If we started with 3 of the ribose 5-phosphates, we would get 2 of these fructose and 1 of the glyceraldehyde 3-phosphate. And just the interesting pieces, 5 carbons times 3, 15 carbons. 6 carbons times 2 is 12, plus 3, 15 carbons. So you can count those carbons and see that they're all accounted for. All right, let's take a look at the oxidative phase of the pentose phosphate pathway. There are 2 oxidative steps. I'm going to highlight those. So this step right here is an oxidative step, and this step. So the first and the third steps are oxidative. All right, so we are oxidizing the glucose 6-phosphate, and we can see, what color do I want? That the NADP goes to NADPH. It's gaining, it's picking up those electrons. Okay, so it's being reduced. So this is being reduced. This is being oxidized. Okay, it's losing electrons. If we look at the second one over here, we see that besides the reduction, right, so it is again a redox reaction, it is also a decarboxylation. So when we say that, we call that an oxidative decarboxylation reaction. Oxidative, there we go. And we can see that, right? You can see that this is breaking off. Okay, we're going from 6 carbon to a 5 carbon. Let's take a closer look at the first step, because that is the step that is rate controlling. So step 1 is rate controlling. The concentration of NADP is really what controls, here we go, the concentration of this is really what controls this reaction. If you have a low concentration, the reaction will not go forward. Okay? There's a couple reasons for that. One is because it's needed for the redox reaction to occur, right? It is one of our substrates, we need that, so it's needed for the reaction to occur. The second thing is that both NADP and NADPH can both bond to the active site, okay, in our glucose 6-phosphate dehydrogenase. So both of those can bond. In a normal cell, you have about 70 times as much NADPH as the NAD+. Okay, NADPH is used in biosynthetic processes. It's typically it's an important metabolite, so we typically have a lot at the ready. Okay, and so since the active site in our enzyme can bond with either of these, and there's 70 times more of this, there is a really good chance that our active site is tied up with this. Okay, and when that's true, our reaction's not going to go forward because what needs to react, right, is our glucose 6-phosphate and the NADP+. So these are going back and forth between each other, right? We have it either in this form or in this form. So as the amount of this NADP goes down, the amount of NADPH goes up. So both, we don't have enough, or we don't have as much of our reactant, and we've got our active sites bound up. As the amount of the NADPH goes down, the amount of this goes up, so we've got more of this, and it's got a better shot at, statistically, like being the one in the active site. Okay, so it's a simple control mechanism dependent on the substrate concentration. And this is kind of our limiting factor. The amount of this will determine our rate of reaction. As we look at steps 2 and 3, okay, we've got a hydrolysis reaction here in the middle. Okay, and then we have our oxidative decarboxylation. So that decarboxylation helps drive this second reaction forward. We've seen that in past processes. So what we want to talk about now is starting with this glucose 6-phosphate, kind of where does it go and what do we want to make from it? So if you go back to the metabolic map, you'll remember there are lots and lots of options, lots of different ways this can go. And it turns out that our pentose phosphate pathway can operate in 4 distinct modes depending on the various combinations of oxidative phase, non-oxidative phase, glycolysis, gluconeogenesis. And our 4 kind of modes are right here. Notice that they are all based on what the cell needs. Okay, and we're going to come and we're going to look at those individually in just a second. So that's why I'm not reading through them all right now. So if you look at it, you'll see those modes listed here, mode 1, 2, 3. In all of these cases, I'm starting up here with glucose 6-phosphate, glucose 6-phosphate, glucose 6-phosphate, glucose 6-phosphate. But where it goes from there is dependent on which mode we're in. Okay, all 4 of these paths look different. So the PPP and glycolysis and gluconeogenesis are sharing a lot of metabolites back and forth. And so just to place ourselves here before we move on to the next slide and look at them in detail, the blue and the green are the pentose phosphate pathway. The blue is phase 1, oxidative. So when you see blue arrows, it's that oxidative pathway. We're going to that ribulose 5-phosphate. When you see green, it's phase 2, the non-oxidative pathways that can take us to many different sugars. When you see orange arrows, now we've dropped into the glycolysis, those original ones we looked at. And then when you see purple, it's gluconeogenesis, building back up towards the glucose 6-phosphate. All right, so now we're going to look, exact same picture, but I've come in and put in the description of each of these modes. You don't need to memorize like mode 1 is this, mode 2 is that, okay? But you should be able to think about what's happening. So for example, our need for this ribose 5-phosphate exceeds the need for the NADPH. So what we really need is essentially this starting material, right? This ribose 5-phosphate. That's mode 1. So when I see that happens, the glucose 6 phosphate is shuttled into the glycolysis pathway. So we see that right here with the orange arrows. And from that, we get down to this glyceraldehyde 3-phosphate. Remember, we saw that in our non-oxidative phase. We get down to these various molecules, and those enzymes, there's a lot of different enzymes in the non-oxidative pathway. Okay, I'm going to scroll for a second so we can go back and look at that. So lots and lots and lots of different enzymes that interconvert through all of these things. Okay? The reactions are reversible. They can go in both directions. And so in this case, we're going to use, we don't want to drop our glucose 6-phosphate into that PPP oxidative phase. That was to get the NADPH, and we don't need a lot of that. What we need are these material needs of our cell met. So we're going to drop through glucose to get to a couple of these. I'm sorry, not glucose, glycolysis. All right, and then, remember the green was the non-oxidative phase. We're going to work our way back up to that ribose 5-phosphate, okay, through those, with those enzymes from phase 2. So that's one option. But another option that you might have is mode 2 over here. So the NADPH and ribose 5-phosphate needs are balanced. We need both. And so what happens here, if you look at it, we've got our oxidative phase, gives us our NADPH. It gets us to this ribulose, and then that isomerase takes us down to the ribose. So essentially what we're going to do is we're going to stop after the oxidative phase. Okay, so for mode 2, we're really stopping mainly after the oxidative phase because we've made both the NADPH and this ribose 5-phosphate, which we want. So we don't want to interconvert it. Okay, an example that when your body might be in this mode 2 would be when you have cells rapidly dividing. When that happens, we need lots of our basic material to synthesize our nucleic acids, and we need that NADPH to power that biosynthesis. All right, mode 3. Now we need more of that NADPH than the ribose 5-phosphate. So our balance is different now. In that case, we know we're going to want to go through the oxidative phase. So here we go. We've got our glucose 6-phosphate. We're going through the oxidative phase to make this NADPH. Our isomerase drops it down to the ribose. But we don't really need that ribose. All right, and so what we're going to do is essentially shuttle it backwards through these non-oxidative phases to other metabolites from the gluconeogenesis. So we can essentially recycle those carbons back up to glucose 6-phosphate and do this again so we can get more NADPH. So an example when you might have this situation might be in your liver cells when you're synthesizing fatty acids. So you don't need the ribose. We're not synthesizing nucleic acids. But we need a lot of this reducing power because we're essentially storing up energy in these fatty acids. Okay, cholesterol synthesis also requires quite a bit of the NADPH. All right, and mode 4. Now we need the NADPH and we need ATP. All right, in this case, we need the NADPH, so we know we're going to go through this oxidative phase. our isomerase drops it down to the ribose, but you know what? We don't really need the ribose. So we can go through those non-oxidative routes, those reversible reactions, and create metabolites, fructose 6-phosphate, glyceraldehyde 3-phosphate, that run through glycolysis. And so we can make the ATP using that glycolysis. Then you also know we're going to make the pyruvate, so we could drop that down into the citric acid cycle. So the big idea here is that this PPP, this pentose phosphate pathway, has multiple functionalities, tightly associated with glycolysis and gluconeogenesis. Okay, the pathway for PPP, glycolysis, gluconeogenesis, the pathways will depend on the needs of the cell. Some tissues, you don't have to memorize these, but just ideas. Now, PPP is active in all living things. It's incredibly important. These are in human, okay? And so you can see that tissues that have active pentose phosphate pathways are used for steroid synthesis, fatty acid synthesis, maintenance of reduced see if I can speak today, glutathione, I can't talk. Let's take a look at that one, because this is kind of an interesting application. So glutathione is made up of a glycine, a cysteine, and a glutamate. That's a little bit interesting. So if you look at this, let's see, right here was the alpha carbon from my glycine. Okay, right here was the alpha carbon from the cysteine. So you can see this peptide bond right here. But then if you come up here and you look at this, it looks a little weird. And that's because the second peptide bond is actually not made with the alpha carbon, but instead it is actually between the R side chain. Okay, so the peptide bond between the glutamate and the cysteine is done with that R side chain of the glutamate. All right, so it's a little bit different. So why do we care about this? Essentially, in your body, you've got a bunch of oxygen. Oxygen is really important for us. In cellular respiration, there are side reactions that create reactive oxygen-free radicals. So we can get peroxides. You can get molecules like this, very reactive molecules with unpaired electrons. In your body, you also have quite a few antioxidant molecules that have antioxidant properties so that they can help deal with these. Glutathione is an important one. So glutathione can come in and react with these species and donate. Let me get a different color here. Oh, that's not going to work. Here we go. Donate electrons from this bond. Okay, when it does that, it forms a disulfide bond, so we get a dimer. It's got two of these glutathiones attached with a disulfide bond. But it can essentially neutralize these by donating electrons. So neutralize these free radicals by donating electrons. But it only works when it has these electrons here. It does not work when it is dimerized, right? So we had our sulfur and then it would be bound to another sulfur. So you'd have two of them. Well, there's no electrons that it has available to give away. So this is the reduced form, and we're going to give away that those electrons, and now it will be the oxidized form, right? We're going to oxidize it, give away those. We need to get those electrons back. And what provides those electrons is that NADPH. Okay, so that NADPH that we get from the pentose phosphate pathway is what provides those electrons. All right, if we think about a common genetic disorder, it is actually a disorder of this particular enzyme. This enzyme is really important in red blood cells. Okay, other cells have other ways of making NADPH, but in red blood cells, it is only the pentose phosphate pathway that provides this NADPH. So in your red blood cells, you have, well, let's talk about the disorder first. As we're talking about, this is actually the most common inherited genetic disorder, is this disorder of this enzyme. People with the disorder produce lower amounts of the enzyme. Now, if you produce none of the enzyme, you wouldn't make it out of the embryonic phase. Okay, so everybody who's alive can produce at least some of this enzyme. But people who have lower amounts often don't know it, and they only discover it when they undergo some sort of oxidative stress. So, for example, you have a really bad infection, and your autoimmune system produces a lot of reactive oxygen species that are meant to go out and kill whatever causes the infection. Normally, that oxidative stress is contained by this glutathione. Okay, it's going to come in and start neutralizing those excess reactive oxygen species so that they don't damage your cells. All right, but if you don't have enough of this, you can't make the NADPH that we need to regenerate this in its reduced form so it's usable. So for people who don't have enough of this and go through some sort of oxidative stress, the body, your red blood cells just can't deal with it. They'll often break open. It leads to something called hemolytic anemia because your red blood cells have broken open. You can't carry the oxygen. So anyway, the pentose phosphate pathway is an extremely important pathway in your metabolic map here. All right, that is it for this video.